week 6 of our lockdown here in the United Kingdom. I'm Chris Astill-Smith. I'm an osteopath, a naturopath and applied kinesiologist. I'm also Director of Research and Development here at Epigenetics Limited. I've been inspired to write this presentation by the recent excellent webinars given by Wally Schmidt and Kerry McCord, and most recently by Tyrone Mincy from New Jersey. I'm going to call this talk Optimizing the Immune System Against the COVID-19 virus. So let's start by looking at the structure of the virus itself. So here we see the familiar RNA coil of the virus that contains the genetic material. And the points of most interest are the spike protein here, which is made of a glycoprotein, as we can see on the outside, and the envelope, which is made of lipids or oils and protein, which forms the membrane around the RNA nucleus. So to note the two major parts, the spike protein here on another picture with the made of proteins, and the envelope here made of fats or oils and protein. And these are the vulnerable bits of the virus itself where we can help to modulate this with nutritional intervention. So each spike protein consists of 3 components that combine to form a trimer structure with 2 parts or subunits. We call these S1 and S2. S1 and S2. You can think of the spike as a multi-stage rocket, with S1 being the boosters and S2 as a space shuttle. Once attached to the ACE2 receptor, a spike sheds its S1 subunit and the remaining S2 part changes its shape or conformation to enable the virus envelope to fuse with the outer membrane and drop the virus's genetic material inside the cell. So zinc may possibly inhibit the change in this conformation, as we'll see a bit later on. So let's talk a little bit about spectroscopic emissions at this stage. A spectroscopic emission, every living and non-living compound, whether organic or not, emits a spectroscopic emission when heated. In other words, put into an excited state. And in this case, we saw the spectroscopic emission of iron. An example of this, which we see every day here in the UK, is when the orange street lights are turned on at dusk time. These lights contain sodium gas, which has a spectroscopic emission of this. So this is the spectroscopic emission here of sodium, but the human eye sees the orange glow when all these colors come together. So however, when all these different colors are layered together, we as humans see the bright orange glow when the light is on, which can be measured as a single monochromic color. The human eye perceives light, as we know, from the violet end of the spectrum at 385 nanometers to the red end of the spectrum at 645, which stretches continually then to 770 nanometers. All of the human eye sees this as red. My finding is that COVID-19 emits a spectroscopic emission, and this emission is at 633 nanometers in the orange spectrum. So here we see the 633 nanometer spectroscopic emission, and you can print this off either on paper or best on an acetate sheet. If on paper, place its color side down on the patient, or the patient can look at it with their eyes. This acetate is now available from Epigenetics at the website www.epigenetics-international.com. So recently we've been news of specifically trained COVID detection dogs set to sniff out coronavirus. Specifically trained COVID detection dogs could soon be sniffing out coronavirus in infected people in the United Kingdom if a trial by British researchers goes to plan. Canine's acute sense of smell is already used to detect certain cancers and other diseases, and it's hoped that the animals could turn their noses to helping with the ongoing pandemic. Some diseases have a distinctive odor that is not detectable to most humans, but which dogs, with their strongly developed sense of smell, find obvious. Infectious odors are volatile organic compounds emitted in the breath, the sweat, the urine, and the feces. Every chemical compound has a specific spectroscopic emission, and I wonder if this is what the 633 nanometer acetate is resonating with. Thus, if a patient weakens in the clear to the specific colored acetate, you could say that they might have currently COVID-19. But of course, never base a diagnosis on this finding alone. You must follow government guidelines in such a case. Now, common symptoms of the COVID-19, as we know, is fever, fatigue, dry cough, loss of appetite, body aches, shortness of breath, mucus or phlegm. Now, in addition to this, other symptoms include sore throat, headaches, chills, sometimes with shaking, and more recently accepted is the loss of sense of smell and taste, the stuffy blocked up nose, nausea and vomiting or vomiting, diarrhea, and the skin rashes. I've tested this remotely using hair samples of known cases of COVID-19, and they weaken in the clear if the virus is still active, regardless of whether the patient has symptoms or not. Most people do not weaken in the clear to this acetate, as they're currently not infectious. However, most people who do not weaken in the clear weaken when the acetate is challenged, whilst therapy localizing to the current 1 or mu time point. So a 1 or mu time point is the coupled meridian's alarm point to the now time. So if we take the now time as the small intestine between 2 and 4 in the afternoon, the 1 time would be the heart, in other words, the couple to the small intestine. So the heart alarm point shown here is the 1 time to the small intestine now time, 2 o'clock to 4 o'clock p.m. This does not mean that they have COVID-19, but if they were exposed to it, you can find out what will optimize their immune system against it. Such things like vitamin D, zinc, vitamin C, vitamin K2, monolaurate, probiotics, etc. Using the preceding protocol, you have been able to assess a range of nutrients to support you, your family, and your patient's immune systems. Most of the people I put on the vitamin D, zinc, and initially sodium ascorbate have so far not contracted the virus. I've had several people who have tested positive, had mild symptoms, and recovered within a few days. But there appears to be other aspects to the reason why some infected people are affected worse by the virus than others. And one of these is that patients who do not weaken in the clear, but only with therapy localization of the 1 time, do weaken when challenged firstly with genetically modified soy, followed by the 633 nanometer acetate. The genetically modified soy itself does not weaken in the clear, but when left on and then the acetate added, they do weaken. If a person weakens in the clear to the soy, then they probably have an intolerance to it anyway. So here we see the genetically modified soy and the 633 acetate weakening in the clear. So this may mean that the genetically modified soy is in some way facilitating the viral entry. And this works like a transmembrane serine protease, which we're going to talk about a little bit later on. Interestingly, organic soy does not create this weakening. 93% of the world's soy is genetically modified, but be aware that much organic is affected also by cross-contamination. Genetically modified soybeans. Following expiration of Monsanto's patent on the first variety of glyphosate-resistant Roundup-ready soybeans in 2015, development began on glyphosate-resistant generic soybeans. Monsanto developed a glyphosate-resistant soybean that expresses Cry1AC protein from Bacillus thuringiensis and the glyphosate-resistant gene. Cry1AC is one of the delta endotoxins produced by this bacterium, which acts as an insecticide. Because of this, the genes for these have been introduced into the commercially important crops by genetic engineering, such as soy, cotton, and corn, in order to confer pest resistance on these plants. Cry1AC is also a mucosal adjuvant, in other words, an over-immune response enhancer for human beings. And it is also an inhibitor of mitochondrial respiration. Cry1AC is a toxin that would have to be detoxified if ingested in humans, most likely by conjugation with glutathione. All my patients positive to COVID-19 show low in reduced glutathione and underexpression of glutathione S transferase of the microsomal type of enzyme. Here on the right, we see glutathione composed of the 3 amino acids, glutamic acid, cysteine, and glycine, And of course, glutathione is the main intracellular antioxidant and detoxifier. So here we see the arachidonic acid cascade going through the lipoxygenase enzyme to leukotrienes A4 here, leukotrienes A4. So the membrane-associated proteins in eicosanoid and glutathione metabolism family consists of 6 human proteins, which includes the microsomal glutathione S transferase enzyme. And these are involved in the production of leukotrienes and prostaglandins E series, both important mediators of inflammation. This gene encodes enzymes that catalyzes the conjugation of leukotriene A4 and reduced glutathione to produce leukotriene C4. This enzyme also demonstrates glutathione-dependent peroxidase activity towards lipid hydroperoxides. This shunting of the leukotriene A4 to the formation of B4 if we have insufficient glutathione or an underexpression of the glutathione S transferase is the most inflammatory of all chemical mediators, especially to the lungs. So this pathway here is inhibited by the toxin, the Cry1AC toxin, due to a deficiency of glutathione or an insufficient production of glutathione S transferase. Leukotriene A4 is then shunted to leukotriene B4, which is the most inflammatory of all chemical mediators. Leukotriene B4's primary function is to recruit neutrophils to the areas of tissue damage, though it also helps promote the production of inflammatory cytokines by various immune cells. So neutrophils, as we know, generate hypochlorite radical via the enzyme myeloperoxidase. Drugs that block the actions of leukotriene B4 have shown some efficacy in slowing the progression of neutrophil-mediated diseases. So leukotriene B4 is a leukotriene involved in inflammation. It is produced from leukocytes in response to inflammatory mediators and is able to induce the adhesion and activation of leukocytes on the endothelium, allowing them to bind to and cross into the tissues. Cytokine storm syndrome is a form of systemic inflammatory response syndrome and is an adverse effect of some viral infections, but also chemicals and possibly some drugs. A number of deaths due to COVID-19 have been attributed to the cytokine storm syndrome. I thus strongly advise all people to strictly avoid all soy products in their diet. Using the preceding challenge, you may find a need for supplementing extra reduced glutathione into the immune system, optimizing the regime along with, of course, vitamin C, zinc, and vitamin D. So here we see glutathione, and the most biologically available is the liposomal glutathione. 5 ml rubbed onto the skin will deliver 400 micrograms of reduced glutathione. So soya products, which we know contain soya, is soya protein isolates, soya milk, soya cheese, soya ice cream, soya yogurt, soya flour, tofu, miso, natto, soyu, soy sauce, tamari, idamadan, soya vegetable oils, tempeh, vegetable gums, vegetable broth, vegetable starch, hydrolyzed vegetable protein, textured vegetable protein, soy molasses, hydrolyzed plant protein, monosodium glutamate, artificial flavorings, natural flavorings, so-called plum sauce, hoisin sauce, fish sauce, teriyaki sauce, instant gravy granules, bouillon cubes, okara, soya bean butter, vegetable gum, gum arabic, guar gum, vegetable starch, soya lecithin, some cosmetics and lip balms, bean sprouts, mono and diglycerides, thickeners, and mixed tocopherols, those vitamin E made from soya. These products may contain soya products, candy, cereals, Asian foods, baked goods and baking mixes, chicken processed with chicken broth, chocolate, deli meats made with hydrolyzed vegetable protein, energy bars or nutrition bars, hamburger meat with soya protein fillers, hamburger buns made with soya flour, ice cream, imitation dairy foods, infant formulas, margarines, mayonnaise, nutritional supplements, peanut butter and peanuts butter substitutes, commercial sauces, gravies and soups, sweet bean sauce, sausages and hot dogs made with soya protein fillers, smoothies, vegetable broth and vegetarian meat substitutes. Interestingly, vitamin C aids in the recycling of glutathione, which we saw was important to detoxifying the Cry1AC. Zinc cofactors the conversion of cysteine into glutathione and activates the ACE2 enzyme. And vitamin D stimulates the synthesis of glutathione and reduces expression of gamma-glutamyl transferase. And here we see gamma-glutamyl transferase, this enzyme that sits on the cell membrane, which breaks down glutathione into the 3 component amino acids. And when this level is high, we break down glutathione, so the body has to make it inside the cell, which is a very energy-dependent mechanism and not so efficient. And this is inhibited by vitamin D. So gamma-glutamyl transferase, GGT, is inhibited by vitamin D. So vitamin D allows more glutathione to get into the cell from the diet. So here we see glutathione itself, and on the left here, the glutathione recycling, glutathione peroxidase, glutathione reductase. And on the right here, we see the microsomal glutathione S transferase, which is this one at 370 nanometers. But there's a whole range here of family of glutathione S transferase enzymes as well, in addition, which are there mainly to detoxify various xenobiotics and toxic metals. It's interesting also to note that in the UK, Britain's ethnic minorities, especially the black people, especially those from the Caribbean, Asians, and also obese people, are suffering a vastly higher proportion of COVID-19 deaths. Could this be due, at least in part, to their diet, lack of vitamin D, and possibly here soya intake in one form or another? And 1/3 of all UK deaths have been attributed to diabetes as a comorbidity. And as we know, diabetes is usually associated with chronic zinc deficiencies. So let's have a look at some treatment approaches now. So we listed these as 4. So number 1, blocking the entry of the virus from docking onto the ACE2 receptor. So here we want to stop the virus from attacking through the spike protein onto the ACE receptor. And as we'll see, zinc, and we find that zinc ascorbate and zinc sulfate are the most biologically active forms of zinc in this particular case. And we'll see that the bioflavonoid quercetin has a big role to play here in delivering zinc inside the cell. And monolaurine from coconut, as we'll see, has a very important part as a surfactant to destroy the envelope of the virus. Number 2, blocking the machinery of the virus, i.e. stopping its ability to replicate. And here again, zinc, as ascorbate and sulfate, has been shown to inhibit the ability of the replication of the virus. Possibly, maybe colloidal silver could have an effect here, and selenium has a long track record of inhibiting viral replication. For the COVID-19 virus to reproduce, once it enters into the cell, an enzyme called RNA-dependent RNA polymerase makes more copies of the virus. However, zinc inhibits this enzyme, and at high enough concentrations within the cell may entirely inhibit the virus from replicating. Number 3, blocking the packaging of the virus. So here we see the packaging of the virus inside the cell. And maybe this will be where medicinal intervention, natural intervention, such as Artemisia annua, cinnamon, echinacea, garlic, pau d'arco, and maybe many other herbs and spices can have an effect here also. Number 4, kill the factory of the virus, in other words, the infected cell itself, by increasing the production of natural killer cells and killer T cells. And here undoubtedly we cover a role for vitamin D, as we'll see in its activated form, vitamin K2, especially the K2 in black cumin seed oil, zinc, and garlic, because it's known that garlic increases nitric oxide. So immune clue sparks treatment hope. UK scientists are to begin testing a treatment that it's hoped could counter the effects of COVID-19 in the most seriously ill patients. It's been found that those with the most severe form of the disease has extremely low numbers of an immune cell called the T cells. T cells clear infections from the body. The clinical trial will evaluate a drug called interleukin-7, known to boost T cell numbers and can aid patient recovery. There's some evidence that echinacea, as Echinacea purpura, can do this. So maybe this is the mechanism that echinacea works as an antiviral natural remedy. So let's start with the ACE2 receptor itself and the relationship to zinc. So angiotensin converting enzyme, or ACE2 receptor, serves as the entry point into the cell for some coronaviruses, and specifically the COVID-19. ACE2 is an enzyme attached to the outer surface, or in other words, the cell membranes of cells, to type 2 alveolar cells in the lungs, the arteries and the veins, the heart, the kidney, and the small intestine enterocytes. Recent research has also shown receptors in the nasal passages and the tongue. So here we see the picture on the right here, the arteries, the lungs, the intestines, and as we'll see, the brain and the heart and the kidneys all have ACE2 receptors. And here we see the possibility of the new finding that the ACE receptors are rich in the nostrils of the nose and also on the tongue. Now, the ACE2 receptors are also found in the cerebral cortex, the striatum, the hypothalamus, and the brain stem within the central nervous system, and could link with the symptoms of delirium with severely infected people who end up on ventilators. As it's known, the severe people who end up on ventilators often have delirium for some time after coming off the ventilator. The expression of ACE2 in cortical neurons and glia make them susceptible to COVID-19 attack, which was the possible basis of the loss of smell, or anosmia, and incidences of neurological deficits seen in COVID-19. And these neurological disorders can continue, as we'll see at the end of the talk, for many weeks or months afterwards. And this article says that young women are more likely to suffer new corona symptoms of loss of smell, doctors find. So it's curious because we find that zinc is also needed for your sense of taste and smell. Because one of the enzymes crucial for proper taste and smell is dependent on this nutrient, a zinc deficiency can reduce your ability to taste and smell anyway. It may be that the symptoms of loss of smell and taste arise because all the zinc inside the infected cells in the nostrils and taste buds has been used up by these cells in fighting the viral infection. ACE2 lowers blood pressure by catalyzing the hydrolysis of angiotensin II, a vasoconstrictor peptide, into angiotensin 1-7, which is a vasodilator. So here we see angiotensin II and angiotensin 1-7. Angiotensin converting enzyme 2 counters the activity of the related angiotensin converting enzyme, in other words, ACE1, by reducing the amount of angiotensin 2 and increasing angiotensin 1-7. So angiotensin 2 going to angiotensin 1-7. So angiotensin 2 is a vasoconstrictor, oxidative stress, cell proliferation, fibrosis, which of course is very important, finding in serious cases of lung pathology with COVID-19, and inflammation, pro-inflammatory. Whereas angiotensin 1-7 is vasodilatory, antioxidative, antiproliferative, antifibrotic, and anti-inflammatory. So ACE2 is a zinc-containing metalloenzyme located on the surface of endothelial cells and other cells. So here we see the zinc attached to the ACE2 enzyme. So interestingly, all these enzymes, conversion here of angiotensinogen to angiotensin 1, which is stimulated by renin, as we'll see in a minute. Angiotensin 1 is converted to angiotensin 2. So the ACE or ACE1 enzyme is zinc dependent. The conversion of angiotensin 2 into angiotensin 1-7 The ACE2 is zinc dependent, and the alternative route of angiotensin 1 to angiotensin 1 to 7 by thimet and neprisin are both zinc dependent also. So zinc, cofactor in the ACE2, is necessary to attach to the negatively charged glutamic acid amino acid in its structure to change its conformation of the protein from an inactive form to an active form. So we know that glutamic acid has a negative charge to it and zinc has a positive, two positive charges. And when the positive attaches to the negative, here they come together, it changes the shape or conformation of the protein. In other words, it activates it. So angiotensin 1 to 7 has been shown to have antioxidant, anti-inflammatory effects. It plays protective roles in cardiomyocytes of spontaneously hypertensive rats by increasing the expression of endothelial and neuronal nitric oxide synthase enzyme, leading to augmented production of nitric oxide. So nitric oxide, along with peroxynitrite, when nitric oxide combines non-enzymatically to superoxide, is the main killing mechanism to kill the COVID-19 virus by the immune system. So we see here, these are the arginine or nitrogen reactive species family. So COVID-19 virus enters human cells via the ACE2 receptor. Viral particles bind to the ACE2 receptor and together they travel into the cell. These viral particles combine to a large number of ACE2 molecules, sequestering them from the cell surface and decreasing ACE2 even further. The accompanying loss of ACE2 function can cause serious health consequences due to ACE2's participation in key physiological processes, main one being regulating blood pressure. So deficiency of vitamin D leads to an overexpression of renin, an enzyme produced in the kidneys. So here we see the kidneys producing the enzyme renin when there's a fall in blood pressure. So a fall in blood pressure stimulates the kidneys to produce renin, which then converts angiotensinogen into angiotensin 1. And the subsequent activation here of the renin-angiotensin system, the critical regulator of blood pressure, inflammation, and body fluid homeostasis. Loss of ACE2 function in the setting of the COVID-19 infection upsets the balance of this critical system, promoting neutrophil infiltration, excessive inflammation, and lung injury. If lung injury progresses to hypoxia, the kidneys release renin, setting up a vicious cycle for decreasing ACE2 even further. And we know that a lot of COVID-19 patients end up with kidney function dysfunction later on. In turn, lower levels of ACE2 promote more damage, culminating in acute respiratory distress syndrome, which is a severe form of acute lung injury that occurs in as many as 17% of all COVID-19 cases and can lead to respiratory failure and ultimately to death. So zinc decreases and stalls viral replication. Zinc is a charged particle, it has two pluses, and lipid membranes do not allow charged particles to enter the cell. Thus, to get zinc inside the cell, we need a carrier or an ionophore, which is required, and quercetin is a natural zinc ionophore. The best compounds to get zinc into the cell and to activate the ACE2 enzyme would therefore be quercetin, which is the natural zinc ionophore, plus one or two forms of zinc. And we find clinically that zinc ascorbate, because of this ascorbate with the vitamin C, is important, and zinc sulfate. So combating viral infections with zinc and quercetin. A South Korea research paper has demonstrated in vitro that by increasing the zinc concentration in cellular cytoplasm, that viral replication is inhibited. As intracellular levels of zinc are increased, the inhibition of viral replication will reach 100%, according to charts within the paper. The researchers used two anti-malarial drugs, which are ionophores. Ionophores are molecules that can carry a charged ion, like zinc, across a cellular membrane. South Korea has been treating high-risk, critically ill COVID-19 patients with the drug hydroxychloroquine. It is likely the single clinical reason that South Korea has the lowest death rate in the world for COVID-19 victims. Hydroxychloroquine is a pharmaceutical drug that requires a prescription and can have cardiac side effects. However, there's a nutritional supplement called quercetin that is a zinc chelator and an ionophore and requires no prescription. Quercetin has a well-established benefit to lung health, which may be important when combating a lung infection like COVID-19. Quercetin is a flavonoid, specifically a flavanol, from the polyphenolic group of compounds found in food and essential for human health. It's rich in foods such as, and the richest is capers, has twice as much quercetin as in any other food, lavage leaves, elderberry juice, radish leaves, wild rocket, dill, coriander or cilantro herb, fennel leaves, and red onions, particularly the cipolla rossa from southern Italy or Tropea area, in addition to the skins and the pith of citrus fruits. Some flavonoids, like quercetin, are broad-spectrum antivirals. Quercetin has a number of beneficial processes in the body. It's not just a zinc ionophore, it's also an antioxidant, a protein kinase enzyme inhibitor, which we're going to talk about, and an estrogen receptor activator. The ionophore mechanism responsible for the accumulation of intracellular zinc occurs because the cytoplasm is slightly acidic, and the lysosomes are very acidic. The quercetin carrying zinc is slightly alkaline, so once inside, they dissociate, leaving the zinc trapped inside to accumulate inside the cell. Chelators can both remove a metal into or out of the body. An ionophore binds to zinc and carries it past the cell membrane, bypassing the normal zinc transporter. So quercetin fills up the cells and channels that the COVID-19 uses to inflame intracellularly and also inhibits serine protease reaction used by COVID-19 for mass replication. So here we see the interaction between proteins and ACE2 receptor is clearly more complicated than a simple lock and key relationship. Many more molecules may be involved in the process, allowing SARS-CoV-2, in other words COVID-19, to invade cells. At the moment, we know of at least one other key player, which is a transmembrane serine protease 2. You can think of this as an inside man working for the factory that the burglar wants to turn into a robot manufacturing plant. It meets the burglar outside the building to prepare or prime the lockpick, as the spike on the virus, so it will properly fit the factory's locks. This is probably the method quercetin uses to inhibit serine protease. Biologists at the German Primate Institute in Gotham found that COVID-19 depends on this protease to invade cells, or more importantly, from a therapeutic perspective, shows that a protease inhibitor previously proved for clinical use, chemostat mesylate, can block the virus from entering the cells. So it's probable that quercetin has the same effect. In the analogy, the inhibitor is a security guard who intercepts the inside man before they prepare the burglar's lockpick. Serine protease play a vital role in host cell virus fusion activities by priming the virus's spike protein to show the protein named fusion protein. So what would be better than supplementing zinc and quercetin together to elevate intracellular zinc levels in order to inhibit viral replication? Quercetin dihydrate has been shown to be very effective clinically against many forms of viruses in addition. So recommended doses to optimize the immune system. 1 to 2 capsules. Each capsule contains 20 milligrams, so somewhere between 20 milligrams and 40 milligrams a day, which would contain 333 milligrams of quercetin in 1 capsule, so 666 in 2 capsules of quercetin, to be taken first thing in the morning, and here it's important on an empty stomach. Do not consume anything but water for at least 1 hour. This is essential for quercetin to act as an ionophile. So most of us are a little bit wary about taking zinc on an empty stomach, but I've done this for some weeks now, in fact several months now, of taking 2 capsules first thing in the morning on an empty stomach with a glass of water, and have had no nausea or upset stomach as a result. If you take quercetin with the zinc and food, in other words, you take it with a meal, the quercetin will combine with other minerals and everything except the zinc. By taking them together on an empty stomach, you're ensuring that the quercetin and the zinc combine and are absorbed into the blood plasma. So recommended doses during an acute viral infection: 2 capsules, again supplying 40 milligrams of zinc and 660 milligrams of quercetin, could be taken up to 3 times a day, leaving at least 5 hours between doses to be taken on an empty stomach. Do not consume anything but water for at least an hour after each dose, and take this dose for 3 to 5 days only. Quercetin has an anticoagulant power which must be taken into account, in particular for people who are already under such treatment medication, because of interaction between anticoagulant drugs and quercetin is possible. So beware if you're already taking or patients are taking anticoagulant drugs. So this is now on the label with the zinc quercetin. So now let's have a look at the immune function role of vitamin D. So vitamin D acts as an endocrine repressor of the angiotensin, the renin-angiotensin system, by downregulating the expression of renin, the rate-limiting enzyme of the renin-angiotensin cascade, and rescuing lung function. So here we see low levels of vitamin D allow renin to be secreted by the kidneys, which stimulates the conversion of angiotensin into angiotensin into angiotensin. Therefore, increasing vitamin D levels will reduce the level and secretion of renin. A preclinical model of acute lung injury showed that administration of vitamin D provided protection against lung injury by increasing the ACE2 levels and decreasing renin production. Vitamin D supplementation increased ACE2 receptor levels, but only in conditions of acute lung injury, where ACE2 levels decreased. When vitamin D was given to control animals, it didn't increase ACE2 levels. This means that the vitamin D normalizes the ACE2 levels in situations only where it is decreased. According to the latest research, Russian research given to me by my good friend, Dr. Tatiana Chernysheva from Vladivostok, GM soy was shown to block vitamin D receptors, and it also increases the body's need for vitamin D. Thus, there is a vitamin D deficiency, and so it cannot work against the COVID-19. She goes on to say, I also pay attention to hemoglobin A, assessing the health status of patients who have been diagnosed with COVID-19 and suffered from severe and very severe symptoms, or developed atypical pneumonia, but have won through to recovery. I found out that the patients showed insufficient IgA levels in tests, as well as on no-so diagnosis. Besides, it was noticed that doctors who showed normal and good IgA levels in the blood test while treating COVID-19 patients didn't catch this virus from them. It is important to keep normal IgA levels for COVID-19 carriers and COVID-19 contact persons and COVID-19 patients with mild and moderate symptoms. Chlorella is a very specific type of algae. Laboratory science shows time and time again that chlorella boosts IgA secretion in the mouth and the mucous membranes. This is critical to build our cells' ability to fight off pathogens that infect us by contracting our nose, eyes, and mouth. So regularly take into your account organic chlorella into your regime. So let's have a look at vitamin D and its role in the immune system. So supplemental vitamin D would be a viable means to increase vitamin D to sufficient levels and potentially reduce the risk of complications associated with COVID-19. And now in the Northern Hemisphere here, we're coming into the summertime. We're getting much more ultraviolet in our sunshine, particularly around the midday, the mid-morning periods, mid-afternoon. And this includes the ultraviolet C areas of about 280, 290 nanometers, which is what's necessary to stimulate the conversion of hydroxycholesterol in the skin to vitamin D. It was first observed that all pneumonia cases have vitamin D deficiency. It was then realized that vitamin D is important for the correct function of the immune system. So here we see the dehydroxy cholesterol being converted to vitamin D or cholecalciferol from the skin. This then goes to the liver, where also dietary intake of vitamin D from fish and certain vitamin D supplements will be stored in the liver, where it's hydroxylated to in the 25 position. So the 25 hydroxy vitamin D is the stored form. This then travels to the kidney and to other cells, where it converts to the 125 dihydroxy vitamin D, which is the one that activates the immune system, which is what we're after today. So vitamin D from the diet or from sunshine, conversion of cholesterol is first hydroxylated in the liver to the 25 hydroxy vitamin D. It's then converted to 125 in the kidney. And this is one of the activated forms. There's several activated forms, but the one we're after, which is involved with the immune system, is the 125 dihydroxy vitamin D. This activation can also occur in many other tissues, it's beside the kidney, and it includes here the white blood cells, very important today. The immune system is divided, as you know, between the innate immune system, mediated by phagocytes. This is the first line of defense, but no antibodies are formed. And secondly, the adaptive immune system, which is mediated by the B and T lymphocytes and produces antibodies, but it may take 7 to 10 days to kick into action. So here we see the classification of the different white blood cells. So the non-specific innate immune system is mostly by the phagocytes. These would be the neutrophils, the eosinophils, the basophils, which are from the mast cells. And we've also got the agranulocytes, which is the monocytes, which convert to macrophages here, and the natural killer cells. Whereas the adaptive immune system is mediated by the lymphocytes, and these are divided between the B lymphocytes here and the T lymphocytes, which are subcategories of T helper cells, memory cells, killer T cells, and suppressor T cells. So the B and the T cells form the lymphocytes, which is the adaptive immune system. So the innate immune system, cells like the macrophages attack the virus. The 25 hydroxy vitamin D enters the macrophage and is converted inside the macrophage to the 125. This is then attaches to the cell nucleus and stimulates the release of certain antimicrobial chemicals, cathelicidin, beta defenses, and NF-kappa B, which requires vitamin D. So cathelicidin causes chemotaxis, thus increasing the immune response. It increases macrophage activity to enhance phagocytosis. It increases vascular permeability, and it increases T and B cell proliferation and their activation. Beta defenses can cause the virus cell membrane through its envelope disintegrating, can create holes in the virus membrane, and the defenses can then enter into the cell, into the virus, and destroy it. And it can create metabolic disruption to the virus itself. NF-kappa B plays a key role in regulating the immune response to infection. NF-kappa B is a major transcription factor that regulates genes responsible for both innate and the adaptive immune systems. Upon the activation of either the T or B cell receptor, NF-kappa B can become activated through distinct signaling components. Now, the adaptive immune system, should the immune system, the innate immune system, not fully eradicate the virus, the adaptive immune system is then activated. It takes 5 to 7 days after encountering a new antigen for the adaptive immune system to reach its full activity. This leads to the activation of the Th1 and Th2 immune cells. The Th1 cells further stimulate the innate immune system by releasing interferon gamma, and Th2 cells stimulate the release of antibodies via the B cells, which is stimulated by interleukin-4 and interleukin-5. When vitamin D is present as the 125, in other words, the activated form, it stimulates the Th2 cells to activate the B cells to produce antibodies. So with optimal levels of vitamin D, we're moving this process more over to the right. So we're stimulating more the Th2 status, which then goes on to produce the B cells, which then goes on to produce the antibodies. So another fat-soluble vitamin of interest in this immune system is vitamin K2. So complex signaling and communication mechanisms at the cellular molecular level are at the heart of the immune response. And vitamin K2 is a critical component that is required for both the activation and regulation of much of the cellular machinery responsible for initiating and executing our immune response. So here we see the most common forms of or sources of vitamin K2 are fermented vegetable products. So K2 can act as a cofactor for some plasma proteins, thereby affecting immune and inflammatory responses, particularly mediated by the T cells. Studies have found links between vitamin K2 levels and diseases, including inflammatory diseases and cancers. Nuclear factor kappa B and K2. There is evidence that K can regulate the activation of NF-kappa B. Other cell and animal experiments have looked at the possible regulation of inflammation by K2 inhibition of interleukin-6 released following endotoxin challenge. These researchers also found that menadione, or vitamin K3, the synthetic form, was capable of suppressing lipopolysaccharide-induced NF-kappa B nuclear translocation and tumor necrosis factor alpha release from at least murine macrophage-like cells. Additionally, in the murine model of acute lung injury, acute respiratory distress syndrome, which occurs in the setting of acute severe illness complications by systemic inflammation, menadione, or vitamin K3, also attenuated the lipopolysaccharide-induced severity of lung injury and suppressed the increased serum tumor necrosis factor alpha level. This occurred concomitantly with the inhibition of the lipopolysaccharide-evoked nuclear translocation of NF-kappa B in lung tissue. So vitamin K2 may be something to consider here as well as a modulator of the immune system, where vitamin D would move that immune system further to the right in the picture that we showed. Vitamin K will have a modulating effect on the expression of NF-kappa B, so we'll hold it to stop it from going too far to the right. Vitamin C and the immune system. So vitamin C, otherwise known as ascorbic acid, is a water-soluble vitamin, which when deficient historically caused the symptoms of scurvy, which allowed people to develop more infections, especially pneumonia, skin rashes, receding gums, and loss of teeth. And here we see a pretty typical vitamin C early stage deficiency with the inflammation around the periodontal areas of the gums. Vitamin C has three functions in the body. It protects the mitochondria in the cells from reactive oxygen species produced by the electron transport between complexes 1 to 3. So here we see vitamin C acting as an antioxidant for the release of free radicals, so the leakage of free radicals, whereas the electrons pass from complex 1 to 2 to 3. Number 2, it protects the phagocytic innate immune cells from the very reactive oxygen species they produce when they engulf bacteria and viruses. These would be the neutrophils, eosinophils, and macrophages. The most potent of the reactive oxygen species produced by these cells is hydrogen peroxide, which vitamin C quenches. So here we see the reactive oxygen species here. First of all, with superoxide being reduced to hydrogen peroxide, being reduced to hydroxyl radical, or hopefully to water and oxygen. But if this pathway is inhibited, then we go straight to water, or in the presence of myeloperoxidase, we may go on to produce hypochlorite radical, which can be very damaging, especially to lung tissue. So vitamin C, in the third function of vitamin C, is essential in the hydroxylation first pathway of the synthesis of collagen. So low collagen production leads to poor wound healing, bone abnormalities, fragility of tissues, bleeding gums, and burst blood vessels. So vitamin C, of course, is very, very important in collagen because collagen is the makeup of the lung tissue, and the lung is the vulnerable part of the anatomy to the COVID-19 virus. So let's have a look at vitamin C and its role in the immune system itself. So vitamin C, as we saw, neutralizes reactive oxygen species. It's a regulator of the immune system, and it also stabilizes cell membranes. And here we see vitamin C, or ascorbic acid, in its reduced form, and then in its oxidized form of dehydroascorbic acid, having taken the electron. Vitamin C distributes readily in high concentrations into immune cells. It has antimicrobial and natural killer cell activities. It promotes lymphocyte proliferation and is consumed quickly during infections. It affects indicating a prominent role in the immune system regulation. Currently, there's no long-term studies available until the September from China about the effects of vitamin C on treating COVID-19. But those studies are mostly by using high concentrations of ascorbic acid intravenously. However, there's plenty of studies about the use of supplemental vitamin C in treating other viral infections, and especially influenza. People who do excessive amounts of exercise are more prone to influenza, the common cold and other infections, probably due to the increase in the amount of reactive oxygen species produced in the energy pathway. Megadoses of vitamin C have been shown to prevent these from occurring. So here we see Sebastian Coe and Steve Ovett, who were two of Britain's finest athletes during the 1980s, and they were always lurching from one infection to another and postponing various race meetings. Fantastic athletes, but forever picking up infections. And if we take a closer look here at the nice smiling face here, but take a closer look and look around the gum tissue here of Sebastian Coe and of Steve Ovett, and we'll see that the teeth and the gums are in bad state, which may be an indication that because of the excess training and athletics that they were doing, they were run down in vitamin C and were bordering line on scurvy. So a study with students where 50% of the group were given megadoses of vitamin C showed that 85% of these did not get colds or influenza. In normal people, extra vitamin C only showed 8% benefit, but 14% in children. I was interested in this picture of students supposedly attending a lecture. This one here is on the phone, this one here is on the phone, and these look as though they're thinking about something totally different. Obviously not chiropractic or osteopathic students. However, when they became sick, they then needed extra vitamin C to protect against the increase in reactive oxygen species produced from the phagocytes and the natural killer cells. Probably the most important, especially with COVID-19, is to protect the collagen of the lung tissue, which we've already talked about. So vitamin C or ascorbic acid is good as a maintenance supplement, but in preventing COVID-19 and other viral infections, we need to keep that vitamin C outside of the cell. And so sodium ascorbate, sodium being an extracellular electrolyte, is probably the better of the different forms of ascorbate. But when the infection, if an infection does occur, and in other words, the virus has gone intracellularly, it's always shown that the people with COVID-19 become very deficient in potassium. And I would then change the person to taking potassium ascorbate because potassium is an intracellular electrolyte rather than the sodium ascorbate. So let's have a look at another interesting remedy, which is monolaurine. Lauric acid is a carbon-12 saturated fatty acid derived principally from coconut. So monolaurine itself is a mixture of lauric acid from coconut extract and glycerin. So monolaurine is a surfactant. That is, it's a compound that lowers the surface tension between two liquids, or between a liquid and a solid. Surfactants may act as detergents, wetting agents, emulsifiers, foaming agents, or dispersants. Soap is a surfactant which destroys the virus envelope, hence the reason for the publicity of washing hands for at least 40 seconds, which takes you that time to sing Happy Birthday or another popular songs. Monolaurine from coconut is known to inactivate lipid-coated viruses by binding to the lipid protein envelope of the virus, thereby preventing it from attaching and entering host cells, making infection and replication impossible. So remember, the envelope of the virus is made of this lipid protein, and the surfactant from monolaurine will help to destroy it. Other studies show that monolaurin literally disintegrates the protective viral envelope, thus killing the virus. So let's have a look at management during an acute viral infection. So the stages of the disease, possibly 18%, maybe more. It's perhaps indicated now that it could be much higher than 18% of positive cases are totally asymptomatic. And people have had antibody tests and show positive to antibodies and yet have never had any symptoms. Mild symptoms, serious symptoms, critical symptoms, and finally death. So the first immune system activation is the innate immune system, as we saw here. So if an asymptomatic person starts to develop a fever, cough, gastrointestinal symptoms, loss of smell, etc., we go from being asymptomatic to developing these mild symptoms. And if the innate system fails, we start to activate the adaptive immune system. And some of these symptoms may develop more serious, into shortness of breath and a need for oxygen. The failure of the adaptive immune system converts a serious case into a critical case, and this may need ventilation and can eventually lead to kidney failure. And this requires intensive care unit in the hospital, so critical, and somewhere between 2 and 5%, although there's great variation from country to country, can be lethal. So during an acute infection, the patient will weaken to the 633 nanometer acetate in the clear. At this stage, they may have symptoms or may be symptom-free. Obviously, it's important that the patient is self-isolating this time, so all testing must be carried out remotely, preferably by using a surrogate and the patient's hair sample. So here we see a hair sample in a polythene bag. How to surrogate using a hair sample. You require a minimum of 6 hairs with the roots. Very important to have the roots because the roots contain the DNA of the patient. In a small sealed polythene bag. Let the patient put the hair sample in. Don't put the hair sample in because you'll contaminate it with sweat from your own hands. Place the bag on the patient along with the surrogate vial. This is a vial. This is necessary to protect the vital energy of the surrogate. You can obtain these vials, surrogate vials, to protect the surrogate from epigenetics. So first of all, check that the surrogate now weakens to the patient's body type acetate. So this would be the red, the green, or the blue. So we're seeing whether our patient, with the hair sample and the surrogate on, that the surrogate weakens with the red acetate, or the blue acetate, or the green acetate. So remove then the body acetate if weakness is obtained. Remove the acetate and proceed as like treating the patient directly. The patient will, of course, be in strength once you remove that body type acetate. Place the 633 nanometer acetate on the surrogate's abdomen, and a strong muscle will weaken if they're carrying the virus in the clear. A challenge for which meridian strength. Then test all the surrogate's muscles on this meridian in the clear. From the strength without the 633 acetate on, all those muscles should test weak. So here what we're doing is we're challenging from weakness around the different alarm points to find out which muscle, which meridian negates the positive virus, the 633 acetate. In this case, it's the liver meridian that negates, the liver 14. So therefore test the pectoralis major sternal, bilaterally, and the rhomboid muscles in the clear without the 633 acetate, but leaving the surrogate hair sample and the surrogate vial on. And here you'll find that from strength that these muscles will test weak in the clear. Any effective remedy should strengthen all muscles if it is 100% effective. If a remedy only strengthens 1 or 2 of these muscles, then it's probably not 100% effective. You must look for a remedy that strengthens all the weak associated muscles. So my findings during the acute stage of an infection is continue the patient with vitamin D, but regularly measure the dose. We don't want to overdose with vitamin D, but we need to keep it to an optimal level. So again, measure it on a regular basis to get the correct dose. Of course, at the moment, with the sunshine and the summer coming, we don't need so much vitamin D from a supplemental point of view, as long as we can, of course, convert the cholecalciferol made under the skin into the activated forms. So number 2, continue the patient on the quercetin with the included zinc ascorbate and sulfate, and measure the dose. Maybe up to 60 milligrams of each, once or twice a day with food during the acute attack. So Dr. Zaleka from New York City recommends 220 milligrams of zinc, elemental zinc, daily for 5 days. So a very, very high dose for a relatively short period of time. But using the zinc with the quercetin, remember we're going to, as an ionophore, the quercetin is going to get the zinc absorbed in the cell and probably needs a much lower dose. So maybe 60 milligrams, once or twice a day. So quercetin is our packet of quercetin again with the zinc and the quercetin. So change at this stage from the sodium ascorbate, remember, to the potassium ascorbate. The patient may need 1 or may need 2+ grams, muscle tested 1 to 2 or more grams, 3 times a day to get the ascorbate intracellularly. Very important. Sometimes when a person changes from sodium ascorbate to potassium ascorbate, I then feel that they've got the viruses going intracellularly. Potassium is an intracellular electrolyte and aids the inside of the cell to be less acidic. All positive tested COVID-19 patients have been found to be potassium deficient. So put the patient onto liposomal glutathione, somewhere between 10 and 25 ml, anything up to 3 times a day to increase the intracellular detoxification and therefore to prevent cytokine storms. Alternatively, if you don't have liposomal glutathione, you can use N-acetylcysteine as a substitute. Test the patient for vitamin K2, and we find the K2 here in the dispersed in black cumin seed oil is the most effective of transporting the K2 into the cells. You can test for monolaurin, which is the combination of coconut with glycerin, 3 times a day. Remember to dissolve the envelope of the virus, and to use chlorella regularly to improve the immunoglobulin A. And probiotics. Leading scientists urge UK government to re-examine the gut's role in coronavirus. Some people say that a lot of problems related to immune system function start from malfunctions in the gut. 90% of the immune system is centered around the gut, the moat, and inside the lymphoid tissue of the gut. In this particular article, they recommend taking probiotics, but unfortunately, there's no work on exactly which probiotics. You need to muscle test for the most suitable types of probiotics, because you don't really want to take probiotics unless you're deficient in them. So if you take too much of a probiotic when you don't need it, you may cause an imbalance in it. But certainly probiotics have a major role in the immune system. So keeping social distancing 2 meters apart, wash hands anything up to 8 times a day or more, depends on the work. Wear a face mask, use disposable gloves, disinfect all touched objects, including keys, car keys, doorbells, delivery and credit cards. Here we see Shake Boy Shake. Are you insane, says the dog. So long-term impacts of COVID-19. This is from the weekly magazine The Week on the 25th of April. Becoming increasingly clear that COVID-19 doesn't just affect the lungs, raising concerns that some people who recover from the disease will be left with lasting health problems. A study of 214 hospitalized patients in Wuhan, China, published in the JAMA Neurology, has found that more than a third developed neurological symptoms, including dizziness, headaches, impaired consciousness, skeletal muscle injury, and in a few cases, seizures and strokes. These may have been a result of the virus infiltrating the patient's nervous system, but they could also have been due to the immune system overreacting. The second study in Wuhan found that even when hospitalized patients recovered from the virus, their livers and kidneys showed signs of impairment. COVID-19 is not just a respiratory disorder. Dr. Harlan, a cardiologist at Yale University, told the Los Angeles Times, it can affect the heart, the liver, the kidneys, the brain, the endocrine system, and the blood system. Separately, statistics show that 91% of the people who died with COVID-19 in England and Wales in March had at least one pre-existing health condition. However, in 86% of cases, COVID-19 was the underlying cause of death. This is something which we've got to be ready and prepared, is how do we treat patients who've had the COVID-19 and are supposedly on the way to recovery, but have still got symptoms or have developed symptoms later on as post-viral symptoms, which may be very severe symptoms and last for a long period of time. So obviously, we're looking at post-infections and recuperation. Obviously, the predisposing factors that were present for the virus to infect will still be there. So whatever the situation with the person's health was before they got the virus will be there and maybe worse because of the viral infection. So number one, must get people out into the sunshine to promote the vitamin D and exposed to broad spectrum health giving wave bands of light. So it's not just the ultraviolet that produces the vitamin D, it's other wave bands of light, which are also very important and health giving to the immune system. But as we head into the summer, the incidence of the virus infections, certainly in the United Kingdom, is decreasing rapidly. But we've got to be prepared that this could come back for a second wave when the levels of vitamin D start to drop, roughly 3 months after the last high dose of the vitamin of the ultraviolet, which will bring us into about November to December time, when vitamin D levels will be again rock bottom. Use broad spectrum lighting inside homes. So we need to not use artificial lighting. We need to get broad spectrum lights, which has the full range of the lights, including the early part of the ultraviolet. Regular exercise is known to boost the immune system. Avoid high fat and sugar in the diet. Two things in animal studies that have inhibited the immune system more than anything else is a high fat and high sugar diet. Avoid soy in all its forms for the reasons I stated. Eat plenty of organic fruit and veg because these have more of the bioflavonoids, the vitamin C, etc. in there. And eat whole grains because these contain the full range of the vitamin Bs and don't deplete the system and don't give a glucose surge. Take 1 to 2 capsules of the zinc quercetin each day. This is ongoing. Not just during the season or the period of time that we have the pandemic, but certainly with the autumn and the winter coming up, we need to take and be prepared to keep our immune system very, very good in the coming months. So take zinc quercetin every day. Take 1 to 2 multiple vitamin mineral capsules. This is in people who've generally had the infection and are trying to recover. So they're often depleted in a whole range of different minerals and vitamins, not just the zinc. Take a probiotic, but get this tested as to which is the optimal form of probiotic or probiotics for the patient. Take an omega-3 oil or a flaxseed oil to supply the omega-3 as an alternative to fish oil every day to optimize the cell membranes. And take at least 1 gram of vitamin C capsule every day. So here we see old people's homes which were devastated by the virus. And if we look carefully here, we see cake and biscuits, bowls of sugar, cups of tea, all the things that people in old people's homes enjoy, but are not doing their immune systems any good. And they're indoors in artificial lighting and probably extremely low in vitamin D because they don't go outside into the sunshine. And they're eating a relatively high fat, high sugar, sedentary lifestyle. So from the Daily Telegraph, 21st of May, revealed the long-term effects of COVID-19 that could go on for months. While the vast majority of those who contract COVID-19 will make a full recovery, there's increasing concern about a small but significant number of patients whose symptoms persist weeks and even months after the first falling ill. These long-term symptoms are often bizarre, say experts, and range from strange pains and fevers to debilitating headaches and lethargy. They can impact those who suffer only mildly from the disease initially, and there may be a link with exercise and the recurrence of symptoms. This is because the cause of the virus, of why the person developed the virus in the first place, still hasn't been treated correctly. And so they are the same when they come over the virus, except for the fact they're more run down and can take many weeks and months to recover. So please check yourself and your family daily, as the situation can change from one day rapidly to the next.
No corona, no corona. The days are getting longer, my cabin fever stronger. All due to bad spit on my couch now. I must sit in my pajamas.